everyone. I am Segura Aldimalam from BS Ed Science 2A. So for today's video guys, I will discuss four main topics, and those are experimental evidence in support of fluid mosaic model of membrane, role of lipid molecules in maintaining fluid property of membrane asymmetry, and the last one is constraints on the motility of membrane molecules. So let's proceed to our first topic, which is the experimental evidence in support of fluid mosaic model of membrane. So when we say evidence, there is a good deal of evidence to support the fluid mosaic model of plasma membrane. So the first evidence we have the evidence in support of mosaic arrangement of proteins. Freeze fracture electron microscopy of the plasma membrane by Branton, 1968. He revealed the presence of bumps and depressions 7 to 8 nanomolar in diameter, which are randomly distributed. As you can see at the figure, it is shown to be transmembrane integral protein particles. For the second evidence, we have the evidence in support of fluid property of lipid bilayer. The mobility of membrane proteins due to fluid property of lipid bilayer was demonstrated by a classical experiment of D. Fry and M. Ediden in 1970. Cell fusion is achieved by the use of some pathogens such as an inactivated parainfluenza virus called Sendai virus. When we say pathogen is a membrane fusion promoting factor such as Sendai virus, lysophosphatides, oleic acid, and an electric field. Hybrid cell or heterokaryon produced by the plasma membrane and cytoplasm of both cells. The two cells are originally labeled with fluorescent antibodies of different colors such as fluorescent green and rhodamine red. As you look at the figure, intermixing occurs as the antigens are dispersed and the two colors become less and less detectable. Then after 40 minutes at 37 degrees Celsius, the intermixing of two colors is complete and the two antigens can no longer be distinguished. So let's proceed for our second topic, which is the role of lipid molecules in maintaining fluid property of membrane. Types of movements of lipid molecules. Lipid molecules very rarely migrate from one lipid monolayer of lipid biomolecular layer. Such type of movement is called flip-flop or transbilayer movement. It occurs once a month for any individual lipid molecule. As you can see at the figure, lipid molecules readily exchange places with their neighbors within a monolayer. This results in their rapid lateral diffusion. Individual lipid molecules rotate very rapidly about their long axis and their hydrocarbon chains are flexible. The greatest degree of flexion occurring near the center of the bilayer and the smallest adjacent to the polar head groups. Role of unsaturated fats in increasing membrane fluidity. Double bonds in unsaturated hydrocarbon chains tend to increase the fluidity of a phospholipids bilayer by making it more difficult to pack the chains together. Role of cholesterol in maintaining fluidity of membrane. Cholesterol molecules orient themselves in the lipid bilayer in such a way that their hydroxyl groups remain close to polar head groups of the phospholipids. The rigid plate-like steroid rings interact with and partly immobilize those regions of hydrocarbon chains that are closest to the polar head groups, leaving the rest of the chain flexible at how it's shown on the figure. And now, let's proceed to our third topic, which is the membrane asymmetry. Membrane asymmetry is the irregular distribution of both lipid and protein molecules in both monolayers of the lipid bilayer. A. Phospholipid asymmetry in plasma membrane. It provides the two sides of the plasma membrane with different biophysical properties and influences numerous cellular functions, the inner half and the outer half. The figure shown the example of human erythrocyte plasma membrane. which is the outer half contains those phospholipids which have more saturated fatty acid chains, while the inner half contains those phospholipids which contain terminal amino groups and less saturated fatty acid chains. And as a result, inner monolayer is more fluid than the outer lipid monolayer. Letter B. Protein asymmetry in plasma membrane. The outer and inner sides of plasma membrane and other membranes do not contain either the same types of equal amounts of the various peripheral and integral proteins, like for example, the erythrocytes plasma membrane. The following are the important properties of some of these proteins. The first one we have spectrin and other cytoskeleton proteins. Spectrin is the principal component of the protein meshwork or the cytoskeleton that underlies the erythrocytes plasma membrane. The figure shown on letter A, one spectrin molecule from human red blood cells. On letter B, schematic drawing of the spectrin-based cytoskeleton on the cytoplasmic side of the human red blood cell membrane. The second property we have the glycophorin. It is a small transmembrane glycoprotein or single-pass membrane protein having molecular weight of 55,000 Daltons and 131 amino acid residues. And the last property we have the bond-tree protein. Like the glycophorin, bond-tree protein has 93,000 Dalton molecular weight. It is a transmembrane protein, but it is a multi-pass membrane protein. For example, its highly folded polypeptide chain, about 930 amino acid long. It extends across the lipid bilayer at least 10 times. It also acts as the anion exchange channels in the membrane. And now, let's proceed to our last topic, which is the constraints on the motility of membrane molecules. In the fluid mosaic plasma membrane, there is not complete and independent freedom of movement for its different component molecules. The mobility of some part of lipid molecules is constrained since that remains tightly bound to some of the integral membrane proteins. Like for example, the mobility of lipid molecules surrounding cytochrome oxidase, an enzyme involved in the synthesis of ATP, are immobilized by the enzyme and makes boundary lipid layer. So let's proceed to the intercellular space. In the tissues of multicellular animals, the plasma membranes of two adjacent cells usually remain separated by a space of 10 to 150 adjacent wide. This intercellular space, shown at the figure, is uniform and contains a material of low electron density, which can be considered as a connecting substance. This substance is found to be a mucopolysaccharide. And that ends my report. Thank you for watching and listening.